Hello teachers and friends, this is SGDB5 and this time we're going to present about the role of CDK or cyclic dependent kinases on the cell cycle. This is the member of SGDB5. And next up we're going to continue with the background of our student project.
My name is Luis Felix Juanda with 3 last digit name 117. I will explain about the background of this project. CDKs is an important protein in our body. CDKs with cyclins will tightly regulate cell division in every phase of cell cycles. CDKs is very important studies topic. One of the reasons cancer and tumor are known to related to the abnormality of cell divisions. Study on the CDKs hopefully can lead into a better understanding and treatment related to the cell cycles.
Okay, my name is Catherine Angela and my student ID number is 210251116. Based on the background of this paper, the problems that need to be addressed are, first of all, what is the definition of a cell cycle? What are the phases in a cell cycle? What is the definition of CDK? How is CDK produced? What is the mechanism of CDK in a cell cycle? How is CDK regulated in a cell cycle? And what are the clinical applications of CDK? Then, based on the problem formulation stated, the objectives of this paper are so that students are able to understand the definition of a cell cycle, the phases of a cell cycle, the definition of CDK, how a CDK is produced, the mechanism of CDK in a cell cycle, how a CDK is regulated in a cell cycle, and the clinical applications of CDK. Also, some of the benefits of this paper is that students are able to understand the cell cycle as one of the basic competencies of this block. Students are also able to understand the definition, production process, mechanism, and regulation of CDK as one of the enzymes of interest in the medical field. And by understanding the clinical applications of the CDK, medical students are more exposed to the medical world and able to manage diseases and treatments regarding this enzyme.
Okay, greetings, doctors and friends. My name is Abel Manuel Shanipar with 3 last digit 118. Okay, so today, now we are on to the definition of the cell cycle. Okay, so the cell cycle is a series of events that occur within a cell as it grows and divides. Cells will spend most of their time in so-called interphases, during which they grow, replicate their chromosomes, and prepare for cell division. The cell then exits interphase, undergoes metasis, and completes divisions. The resulting cells, known as daughter cells, each will enter their own interphase and initiate the neuron of the cell cycle. So, the most basic function of the cell cycle is to accurately replicate the vast amount of DNA in a chromosome and will divide that copy into 2 genetically identical daughter cells. DNA replications occurs in S phase, which stands for synthesis, and then there will be a M phase for metasis, and then there will be a G1 phase or gap 1 phase and G2 phase, gap phase 2. Okay, so on the synthesis, on the S phase, it will last for like 10 to 12 hours and accounts for about half the cell cycle time of common mammalian cells, which after the S phase, there will be an M phase, where the chromosomal segregation and cell division that will occur in the M phase. Okay, and the M phase will take much shorter time than the synthesis phase. Okay, thank you.
Hello friends and doctors, my name is Christine Natalia with the 3 last digit student number 109. I'm here to present the phases in cell cycle. According to Cooper in 2019, human cells divide approximately every 24 hours, thus a cycle. The cell grows stable during the interphase, with most outmost dividing cells doubling in size between one mitosis and the next. However, the mitosis and the cytokinesis phase lasts only about an hour. It means most of the cell cycle happens during the interphase. Interphase is the resting time of the cell. Both cell growth and DNA replication occur during the interphase in preparation for cell division. Three phases are consisting the interphase. The first one is gap 1 phase. During the gap 1 phase, the cell will collect all the building pieces of chromosomal DNA and the associated proteins. The cell will also store sufficient energy for completing the task of replicating chromosome in the nucleus. Therefore, the cell is metabolically active and continuously grows in size, but does not replicate its DNA. Furthermore, the gap 1 phase lasts for approximately 11 hours. The second one is the synthesis phase, or S. The synthesis phase makes the duplication of centrosomes. The presence of the synthesis phase results in the formation of sister chromatids, identical pairs of DNA molecules, which results in the rise of the mitotic spindle. S phase took 8 hours to complete, followed by the G2 phase next. The last phase of the interphase is gap 2 phase, or G2. The G2 phase is the completion stage of the interphase. During this phase, the cell growth will continue by duplicating some cell organelles, dismantling the cytoskeleton, and synthesizing the proteins in preparation for mitosis. G2 happens for approximately 4 hours.
Hello Doctor and friends, my name is Grace Veronica Wijaya with the last digit of student number 1 and 13. Now I will explain about the stages of mitosis, which is the last part of phases in the cell cycle. So mitosis starts with the first stage called prophase. In prophase, each two pairs of centriole will move to opposite poles, which result in the presence of the spindle. The second stage is prometaphase, when microtubules pulls the chromosome into opposite direction. The third stage is metaphase, when chromosomes line up in the middle of the cells. The next stage is anaphase, when sister chromatids separate in opposite direction of the cells. The last stage is telophase and cytokinesis, when a new nucleus forms on each side to create a new pair of identical cells, and the cytoplasm separates into two parts.
Hello doctors and friends, my name is Jovita Theodora Liu with the last 3 digits 114. Now I will present to you about CDK. Cyclin-dependent kinases, or CDKs, are a group of multifunctional enzymes that control cell proliferation, regulate cell cycle, and transcription. There are several types of CDK, for example, CDK1, CDK2, CDK4 and 6, and many more. But the main ones are CDK46 and CDK21. CDK46 works from G1 to S phase of the cell cycle, and CDK21 is named based on when it is found in the cell cycle. CDK consists of catalytic and regulatory subunits. The catalytic subunits help CDK's activations for phosphorylation of protein substrate, while the regulatory subunit is cyclin. Cyclin is the protein that controls the progression of the cell cycle and activates CDK. The examples of cyclin are cyclin D, cyclin E, cyclin A, and cyclin B. It is also named based on when it is used in the cell cycle.
Hello doctors and friends, my name is Madinila Mala with the last 3 digits of my student numbers are 106. and now I will continue the presentation with production of CDK. CDK or cyclin-dependent kinase is one of the types included in the CMGC family along with mitogen activated protein kinase or MAPK, glycogen synthase kinase or GSK, and CDC-like kinase or CLK. CDK has a role in the cell cycle. Other studies say that it first appeared in an early eukaryotic ancestor and there is no specific CDK production phase since it naturally exists in the human body. As a part of enzymes family, CDK is found in all eukaryotes. It is produced as a small protein and also consists of catalytic and regulatory subunit. The regulatory subunit is cyclin, which will bind with the catalytic subunit and activate the CDK.
Okay, my name is Agustin Muradinda Pratiaksa with the last 4 digit as 1 and 12. So now I'm going to explain about the mechanism of CDK in cell cycle. As its name, cyclin-dependent kinase, the CDK is activated by cyclin and together form a heterodimer complex that acts as a catalytic subunit in response to signal to regulate the cell division. As described before, human cell has 20 types of the CDK, but only several of them directly regulate the cell cycle transition such as CDK1, CDK2, CDK3, CDK4, and CDK6, whereas the other type of the CDK will mediate the gene transcription. In cell cycle, most of diploid cells in adult female body tissue are in G0 state, both in quiescence or senescence condition. And if mitosis is required, the mitogenic factor will stimulate the quiescent cells to be involved into the cell cycle. And then the signaling cascade will be continued by CDK4 and or CDK6, which is activated by cyclin D, thereby form a cyclin D CDK4 or 6 complex, which initiates the phosphorylation and inactivation of retinoblastoma protein or PRP. And this inactivated PRP will allow the E2F transcription factors to de-repress and induce the transcription of cyclin A, cyclin B, and cyclin E. Thus, the cell cycle can be continued from G1 to S phase. And in other hand, there is a cyclin C CDK3 complex, which regulates the cell to enter or exit the G0 state and also have role in PRB phosphorylation. During the late G1 phase, active cyclin E will bind and activate the CDK2, which also can phosphorylate more PRB, so that causes a positive feedback to the amount of the active CDK2. And this cyclin E CDK2 complex will initiate the S phase. Next, entering the S phase, cyclin A will replace the cyclin E, thereby form a cyclin A CDK2 complex, which ends the S phase by phosphorylating cell division control 6 or CDC6 and E2F, so that the cell enters the G2 phase. Next, in G2 phase, cyclin A will activate the CDK1 and leads cell to enter the M phase. And last, in M phase, the CDK1 is maintained by cyclin B. Then the CDK1 will be phosphorylated, that causes the breakdown of nuclear inflow, chromosome condensation, and the mitotic spindle assembly. And this CDK1 expression will be decreased as the degradation of cyclin B that allows the chromosome to separate and completion of the mitosis and cytokinesis. And this mechanism will be repeated as over the cell cycles. That's all from me. Thank you.
My name is Nibana Siva with the last digit 121, and I'm going to explain the regulation of CDK in cell cycle. As we already know, CDK needs to bind to the specific cyclin and form a cyclin CDK complex. In the process of carrying out its duty, there are some regulations that might affect this activity. CKI or CDK inhibitors are proteins that inhibits the CDK activity. Up until now, there are two families of CKI that have been identified. The first family is called ENK4. This protein will specifically binds into CDK4 and CDK6 and inhibits its activity. And the second family is called CDK interacting protein. This protein works more effectively as an inhibitor than the first family or ENK4. This protein will inhibit inactive cyclin and CDK complexes such as cyclin E with CDK2, cyclin A with CDK2, cyclin B with CDK1, cyclin D with CDK4, and cyclin D with CDK6.
Let me introduce myself first. My name is Novia Indratmo and the last 3 digits of my student number is 119. Let me continue the presentation into the clinical applications of CDK, which is actually the malfunction of CDK in cancer, one of the most dangerous and common diseases in the world. So what is the relation between CDK and cancer? Cancer is a condition where the cells in your body proliferate continuously and uncontrollably. It is actually caused by the hyperactivation of CDK. As previously discussed, CDK is one of the checkpoints in the cell cycle, and CDK can allow the cells to move into the next phase of the cell cycle. Because there is the hyperactivation of CDK, the cells cannot regulate its own cell cycle, which means there is a cell cycle dysregulation. This is the result of a loss of function in the tumor suppressor genes, usually caused by a mutation. So tumor suppressor genes like the p53 controls the expression of p21, which usually can produce proteins which can inhibit the cell cycle, so it can control the proliferation of the cell. But in case of the cancer cell, the genes doesn't work properly, so there is no inhibition in the cell cycle. That's why the cell cycle keeps continuously going. Let's take a closer look in the cell cycle in cancer. So studies have shown that the cancer cells have a shorter G1 phase than normal cells. That means the cell cycle occurs more quickly. There is also a higher level of CDK activity. This means there is the failure of checkpoints and there is also the failure of the cell cycle to rest. That means the cells does not go into the G0 phase, which is the phase that should be where the cells are not actively dividing. This is what causes the continuous proliferation of cancer cells and what causes the uncontrollable growth.
My name is Gwanet Felicia Awaden with student number 21025111110, and I'm going to explain about the update so far on CDK and cancer. With the concept of hyperactivation of CDK causes cancer, there are currently 48 kinases in the market approved by FDA, and 3 of them as CDK inhibitors for breast cancer. The update so far is CDK4 or 6 inhibitor ribociclib shows good signs in animal trials for melanoma growth, while CDK7 is targeted in a lot of disease, which is small cell lung cancer, peripheral T-cell lymphomas, and triple breast cancer. It is thought by researchers that THZ1 is a great potential for high-efficiency medication for CDK7 inhibitor. There are also potential in CDK5 inhibitor as targeted medication for Alzheimer's disease. This is based on the activity of CDK5, which is high activity in central nervous system development and also neuron movement. But overall and generally, results in most cancer aren't that great. However, there are a success, which is in breast cancer. As I've explained before, there are 3 approved medications for breast cancer by FDA, which is abemaciclib, ribociclib, and palbociclib. Abemaciclib and ribociclib have shown good signs and high efficiency in fighting breast cancer, while palbociclib, accompanied with PPPP inhibitor 6AN, shows apoptosis in breast cancer cells, and palbociclib accompanied with small dose of letrozole also shows improvement. Here are the list of targeted CDK inhibitors: palbociclib, abemaciclib, ribociclib, and trilaciclib is an inhibitor for CDK4 or 6, while dinaciclib is for CDK12, nalciclib for CDK2, flavopiridol for CDK9, and roscovitin for CDK2 and 9. That is all from me. I'll pass it to my friend. Thank you. Let me introduce myself. My name is Zafira Kumala Dewi with the last 3 digits of student number is 122. Conclusion. Cell cycle is a series of events that occur within a cell as it grows and divides. The cell cycle consists of 3 phases. First one is interphase. Interphase consists of G1, S, and G2. Next is M phase or mitosis. It consists of prophase, prometaphase, metaphase, anaphase, and telophase. And the third phase is cytokinesis. But to control proliferation and regulate the cell cycle, there is a group of multifunctional enzyme called cyclin-dependent kinase. Cyclin-dependent kinase is divided into 2 subunits. First one is catalytic subunit that helps CDK activation for phosphorylation of protein substrate, and the second one is regulatory subunit that control the progression of cell cycle and activates the CDK. But since it is naturally exists in the human body, there is no specific production phase of CDK. Cyclin-dependent kinase, together with the cyclin, form a heterodimer complex that acts as catalytic subunit in response to the signal to regulate the cell division. Cyclin-dependent kinase has to be activated by cyclin to form a cyclin-CDK complex with different types and function. In general, cyclin-CDK complex drives the cell to enter different phase according to the cell cycle. On the other side, there are some inhibitors that inhibit the CDK activity, such as INK4 and CDK interacting protein. The dysregulation of CDK can lead to loss of cell cycle control. Lack of cell proliferation regulation can lead to abnormal and uncontrolled growth of tissue, or it can be called as cancer. The cell cycle is way more active in cancer cell, leading to the rapid proliferation of cell due to the failure of cell cycle checkpoint by CDK. Because of the hyperactivation of CDK, basically medication for cancer is CDK inhibitors. For instance, CDK4/6 inhibitors, palbociclib, ribociclib, and abemaciclib are used to treat breast cancer. CDK inhibitory treatment still has a great potential to develop, as there are several types of CDK and many varieties of cancer. That's all from us. Thank you for your attention.
Hello doctors and friends, so my name is Stanislaus Nata, and with the last digit of student number 115, so I'm going to tell you about the suggestion for the student project. Based on the results of this research paper, the following things could be recommended to improve the outcome of further studies. First is getting the comprehension on how the CDKs are able to work and its mechanism in the cell cycle. Until then, for those who might have interest regarding disease or pathological conditions that the CDK might affect, you could gain some information regarding the CDK as a whole unit. For researchers, future studies for the CDK application should be conducted in order to improve the healthcare units, moreover towards the pathological conditions that are affected by the CDKs. So that's all from our SGD. Thank you for listening.